Using human cells, scientists have successfully constructed miniature biological robots. These multicellular bots have the ability to navigate and assist in the healing process of artificially created injuries in cultured neurons. Greetings and salutations to everyone. In today's video, we will be discussing the groundbreaking work of researchers from Tufts University and Harvard University's Wyss Institute. They have successfully developed miniature biological robots known as Anthrobots, using human tracheal cells. These remarkable bots have the ability to navigate and assist in the healing process of simulated neuronal injuries. This discovery marks the beginning of a new era in which patient-specific biobots can be utilized as innovative tools for regeneration, healing, and disease treatment. One of the key advantages of using human cells is the ability to construct these bots from a patient's own cells, eliminating the risk of immune responses or the need for immunosuppressant drugs. Researchers have developed multicellular robots, varying in size from the width of a human hair to the tip of a sharpened pencil, that possess the ability to self-assemble. These remarkable biobots have demonstrated an astonishing capacity to heal other cells. This groundbreaking discovery marks the beginning of a journey towards utilizing patient-derived biobots as innovative therapeutic instruments for the purposes of regeneration, healing, and disease treatment. Building upon previous investigations conducted in the laboratories of Michael Levin, Vannevar Bush Professor of Biology at Tufts University School of Arts and Sciences, and Josh Bongard at the University of Vermont, the current study delves into the development of multicellular biological robots known as xenobots. These remarkable entities, derived from frog embryo cells, exhibit a wide range of functionalities, including the ability to navigate through passageways, gather materials, store information, self-repair, and even undergo limited replication. At the time, it remained uncertain whether these capabilities were exclusive to amphibian embryos, or if biobots could be constructed using cells from other species. The recently published study in Advanced Science, conducted by Levin and PhD student Gizem Gumuskaya, reveals an intriguing finding. It is indeed possible to generate bots from fully developed human cells without any genetic alterations. These bots exhibit certain abilities that surpass the previous observations made with xenobots. This discovery marks a significant step towards unraveling the underlying principles governing cellular organization and collaboration within the human body. Moreover, it raises a thought-provoking question. Can cells be extracted from their natural environment and reconfigured into alternative body plans to fulfill specific functions through intentional design? After years of dormancy within the trachea, human cells were given an opportunity to restart and explore novel possibilities for constructing new formations and fulfilling new functions. Our objective was to investigate the potential of cells beyond their default functions within the body, explained Gumuskaya, who possesses a background in architecture prior to entering the field of biology. By reconfiguring cellular interactions, we can generate fresh multicellular structures, much like how stone and brick can be arranged to form various architectural elements such as walls, archways, or columns. The researchers not only discovered that the cells could produce innovative multicellular shapes, but they could also navigate across a surface of cultivated human neurons in a laboratory dish and stimulate the growth of new cells to fill in gaps resulting from the scratching of the cell layer. Although the exact mechanism by which Anthrobots promote neuron growth remains uncertain, the scientists have verified that neurons did indeed develop in the region encompassed by a concentrated group of Anthrobots, referred to as a superbot. According to Levin, who holds positions as the director of the Allen Discovery Center at Tufts and an associate faculty member of the Wyss Institute, the lab-grown cellular structures we create possess capabilities that surpass their natural functions. Levin finds it both captivating and surprising that unaltered tracheal cells from patients can autonomously migrate and stimulate the growth of neurons in areas of injury. Currently, the focus is on unraveling the intricacies of this healing mechanism and exploring the potential additional abilities of these constructs. Utilizing human cells offers several benefits, such as the capacity to create bots using a patient's own cells for therapeutic purposes, without the concern of provoking an immune reaction or necessitating immunosuppressant drugs. These cells have a limited lifespan of a few weeks, allowing for easy reabsorption into the body once their task is completed. Anthrobots. Anthrobots, while capable of surviving in highly controlled laboratory settings, cannot exist outside of the body. Therefore, there is no concern regarding their exposure or accidental dissemination beyond the confines of the lab. Furthermore, Anthrobots do not possess the ability to reproduce or undergo any genetic modifications, ensuring that they will not evolve beyond the established safety measures. Each Anthrobot originates from a single cell obtained from an adult donor. These cells, which are derived from the tracheal surface and adorned with cilia, assist in the expulsion of minuscule particles that enter the lungs' air passages. We all experience the effect of ciliated cells when we cough or clear our throats to eliminate these particles and excess fluid. Previous research conducted by others has demonstrated that these cells, when cultivated in a laboratory environment, naturally aggregate to form small multicellular structures known as organoids. By manipulating the growth conditions, the researchers were able to prompt the cilia on the organoids to orient themselves outward. As a result, the cilia began to exhibit movement resembling the motion of oars. This observation marked a significant milestone in the development of the biorobotics platform, as it unveiled various shapes and patterns of movement. According to Levin, the next step would involve incorporating additional features into the Anthrobots, potentially contributed by different cells. This would enable them to not only respond to their surroundings, but also navigate through the body, fulfilling specific functions or aiding in the construction of engineered tissues within laboratory settings. With the assistance of Simon Garnier from the New Jersey Institute of Technology, the team successfully classified the various Anthrobots that were generated. They discovered that these bots could be categorized based on their shape and movement, spanning a range of sizes from 30 to 500 micrometers, equivalent to the width of a human hair to the tip of a sharpened pencil. This classification is significant, as it fills a crucial gap between nanotechnology and larger engineered devices. There were various shapes and sizes among the Anthrobots, some being spherical and completely adorned with cilia, while others were irregular or football-shaped with sporadic patches of cilia or cilia only on one side. Their movements ranged from travelling in straight lines to performing tight circles or even combining different types of movements. Additionally, some Anthrobots simply remained stationary and wiggled. The spherical ones, fully covered in cilia, were predominantly the ones that exhibited wiggling behaviour. On the other hand, those with unevenly distributed cilia tended to move forward for longer distances, either in straight or curved paths. In laboratory conditions, these Anthrobots typically had a lifespan of around 60 days before naturally undergoing biodegradation. Gumuskaya, the creator of the Anthrobots, revealed that these remarkable machines are capable of self-assembling in a lab dish. What sets them apart from Xenobots is their ability to take shape without the need for tweezers or scalpels, making use of adult cells, including those from elderly patients, instead of relying on embryonic cells. The scalability of this technology is truly impressive, as swarms of these bots can be produced simultaneously, laying the foundation for the development of a therapeutic tool. In order to explore the potential healing capabilities of Anthrobots, Levin and Gumuskaya devised a lab test. By growing a two-dimensional layer of human neurons and lightly scratching it with a slender metal rod, they successfully simulated an open wound, devoid of cells. In order to guarantee that the gap would be fully exposed to a concentrated group of Anthrobots, they devised a strategy involving the creation of superbots, a cluster that naturally emerges when the Anthrobots are confined to a limited area. The superbots consisted mainly of circlers and wigglers, ensuring that they would remain in close proximity to the open wound without straying too far. In the realm of promoting neural growth, one might assume that genetic alterations to Anthrobot cells would be necessary. However, it is quite astonishing that the unaltered Anthrobots were able to initiate significant regrowth, resulting in the formation of a neural bridge that matched the thickness of the surrounding healthy cells. Notably, in areas where Anthrobots were not present, there was no growth of neurons. Within the controlled environment of the lab dish, the Anthrobot collectives effectively facilitated the healing process of live neural tissue. The researchers have indicated that the bots have the potential for various other applications. These include the ability to remove plaque buildup in the arteries of patients with atherosclerosis, repair damage to the spinal cord or retinal nerves, identify bacteria or cancer cells, and deliver targeted drugs to specific tissues. The Anthrobots have the capability to aid in tissue regeneration by administering pro-regenerative drugs. Gumskaya explained that cells possess inherent self-assembly abilities, allowing them to form layers, fold, create spheres, sort and separate themselves based on type, fuse together, and even move. Unlike lifeless bricks, cells are able to communicate with one another and dynamically construct these structures. Each cell is programmed with multiple functions, such as movement, secretion of molecules, and signal detection. The challenge lies in combining these elements to generate novel biological body plans and functions that differ from those observed in nature. The scientists utilize the adaptable regulations of cellular assembly to construct the bots, which not only aids in their understanding of the natural formation of body plans, but also provides insights into the intricate interplay between the genome and environment in the creation of tissues, organs, and limbs, as well as the potential for regenerative treatments. To support our channel's growth and ensure broader awareness, kindly hit the like and subscribe buttons. This will help us reach more individuals and disseminate valuable information. Thank you in advance.